and he's going to sort of present on the methylome browser called OMB. Anshing, you ready?
Yes. Can you hear me?
Yeah.
Okay. So good afternoon, everyone. I hope you can see my slides. So before I introduce our data browser, I just want to give a brief introduction.
We can't see your slides.
Yeah, we can't see your slides, yeah.
Okay. Sorry, let me try. Oh, I haven't started sharing screen, sorry.
There you go. And you're not in present mode.
Yes, I know.
Okay, great.
Okay, thanks. So I want to give a brief introduction about the data set itself. And the browser I'm showing you today is basically a data visualization for the data, methylation data we generated in this project. So we have this companion manuscript called the DNA Methylation Atlas of the Mouse Brain at Single Cell Resolution that will come out with the flagship paper with the included in the whole BICCN Mouse Brain Atlas package. So as many of you might be already familiar with, that in the BICCN, we have all different kinds of cutting edge technologies to study the mouse brain and define cell types using all different kinds of information. And in our lab, we are particularly interested in the epigenetics diversity and more especially the DNA methylation diversity of the mouse brain of the neuron cell types. So several years ago, we developed a technology called the single nuclear methylome sequencing. And in this project, we apply it into the mouse brain. To get the cells, we first dissect the mouse brain into 600 micron coronal slices, and then we separate each several dissection regions contain certain brain anatomical structures to isolate the nuclei. And then we perform the effect sorting to sort most of the 92% of the cell as from the neuron positive gate, and we also cover 8% from the neuron negative gate. Then the cells is sorted into 384 well plates, each one cell per well, and each undergo bisulfite conversion and the SMC62 library preparation separately. And then we do the sequencing to get the cell methylome. And also the same data is going to our collaborator RenLab at UCSD to apply the single nuclear tech-seq, which to get the chromatin accessibility information from the same tissue. So I think you will also hear their very nice browser and studies from this part. But here I'm only focusing on the methylation data. So in the manuscript we currently released, we have more than 110,000 nuclei from 35 brain dissection regions. And here is basically an overview of where the cell was dissected from, covering almost half of the brain. And to identify cell types in this large data set, we used a three-level iterative clustering analysis. We first combine all the data to identify the cell classes, which includes inhibitory, excitatory, and non-neuronal cells. And then we take each cell class to further separate them into major cell types, and then in each of the major cell types to cluster them into subtypes. And by using this approach, we can get this hierarchical definition of cell types at 3 different levels. And there's also cell type labels and UMAP or t-SNE coordinates associated with each level. So you will see later in the browser that all this information can help us better navigate this large data set from the very broad view to very specific details. And if you and also after we go down the clustering, we have we can annotate the cell location based on where the cell is dissected from. And instantly you can see that some cell types, like the non-neuronal cells, might be shared in most of the brain regions, and many other cell types, especially most of the neurons, might be specific to certain brain structures. And by zooming into details of this data set, we could have some interesting findings about specific cell type and their spatial location. For example, in this red circle I point out is an example that might be of interest. We found that most of the excitatory cell in the hippocampus is clustered separately. However, there is a small population that comes from cortex, but co-clustered with excitatory hippocampus excitatory cells. And by looking deeper into their annotation of brain dissections and their marker genes, especially the marker gene pattern in the Allen in situ database, we noticed that this population, which dissected from cortex, might be this small structure called inducium gyrasium, which is actually a structure that classified into the hippocampus in the Allen Brain Atlas. And the molecular profile also shows that excitatory cells from this structure is very close to the hippocampus, especially the CA2 region of the hippocampus, because they basically co-cluster at the major type level. So that's about defining the cell types using the methylation data. And in addition, following that, basically we want to characterize the cell types using the methylation features. And usually in methylation studies, we can separate the methylation signal into two kinds. One is the canonical CpG methylation, which basically are people talk about the brain. Our lab and many others found several years ago that the non-CpG methylation, which stands for ACA, CC, and CT methylation, is also very abundant and cell type specific. So based on previous studies, we find that there might be two ways to define methylation features to describe cell types. One is that in the cell type in the gene body, especially the cell type marker genes, the CH methylation can be very diverse and cell type specific. So it's actually negatively correlated with cell type. So for example, if in cell type A, we see this gene has hypomethylation across the gene body, this probably means that the RNA expression of this gene is highly expressed in this cell type versus the other where the gene body is hypermethylated. So in addition to the MCH, MCG is also showing cell type specificity in many of the non-coding regions that potentially corresponding to the regulatory elements. So these two kinds of information can be used to annotate our cell types by doing the differential analysis. To conduct this kinds of annotation, we used a method, we used a framework to basically pairwise calculate the differential genes or differential methylated regions among any pair of cell types, subtypes in this case. And we can, for each pair, we can define by performing the test, we can determine certain number of differential methylated genes or differential methylated regions. And we can also further run a motif enrichment analysis on the differential methylated regions to further explore whether certain motifs might significantly enriched in a DMR set of one cell type, which indicates that this motif might be involved in regulating the cell type specificity of this cell type. So all this pairwise comparison is also included in our browser, as you will see later. So using this pairwise comparison and the cell type trajectory or taxonomy we can get by running a hierarchical clustering on the subtypes, we then want to annotate each of the node in this taxonomy. The way we do so is by defining a metric called impact score, basically aggregate the pairwise comparison to each of the node by using the comparison involved in the leaf nodes of this particular node. And by doing so, we can annotate each node with their important differential methylated transcription factors or any other genes, and also motif information, which for the interest of time, I'm not showing here. So another important aspect which is not currently under developing for the browser, but I think it's also important to mention and describe in our paper, is that the most important information found in epigenomic data, in addition to genes, are the regulatory elements information. And in our data set, we can identify more than one, by combining the attack data to predict enhancers, we have identified more than 1.6 million enhancer-like DMRs. And to further study the relationship of these enhancers with their potential targeting genes, we have generated a multi-omic data set using the technology SNM3CC, which can basically generating chromatin conformation and the DNA methylation in the same cell. We generated we're using this technology to generate a data set in the hippocampus, and using the additional chromatin contact information, we could potentially annotate many cell type differential methylated regions or the differential peaks to their certain genes by looking at the loops of the chromatin contact information. And this can help us predict potential targeting genes of some of the very significant cell type specific enhancers. And on top of that, one of the last interesting phenomena we see is that we saw that by combining the cell type definition and their spatial location, we find there are spatial gradients that exist in the cell type as shown, identified by their methylation. And here the example is given from in all the cortical excitatory IT neurons. You can see that the cell types can first separate into layers, and then within each layer, there's clear spatial separation based on their dissection. And also in some relatively homogeneous cell type, like the DG granule cells identified in all the dentate gyrus of the hippocampus, we can see that within this homogeneous cell type, we see the methylation gradient, which has correspondence to their spatial distribution. And we can identify genes and differential regulatory elements associated with these gradients. So for the interest of time, I'm not going in detail into every aspect of this data set, but you can go to the bioarchive version of this manuscript if you're interested. So the main purpose here today is showing you the brain single cell epigenomic viewer we built for visualize this data set. And we organize this browser by basically showing, by building some tools to show every different aspect of this data set, including that we can show the profile of certain genes of their different methylation level at different cell types, or we can show the cell type composition of certain brain regions, or we can show cell types with their brain region distribution and their marker genes. Also, one thing we're still under developing is we can show that the cis-regulatory elements associated with either certain gene or certain cell types. So now I will give you a demo to our data visualization browser. Let me also copy the URL to the chat. So in the home page, you can see that there are currently there are three different components of browsing this data set. It's either by gene that you can input a gene name or like get1, get2, or COX2. And then click the go will take you to the gene page. And you can also select a certain brain region to view the data associated with this region. And also you can click on the cell type drop down to select any cell type that we define in the manuscript to see the cell type view. So I will show them one by one. So let's first go to the gene page that I already opened. Here is the page for COX2. So we start with having a brief description of gene card of the basic information related to these genes, and including external database links, especially for some of the gene, the link to the Allen in situ database to see the in situ data in mouse brain. And then on the right side is the summary box plot of this gene's methylation level at different cell types. And as I said, we have defined the cell types at different cluster levels. So, so you can view this data, this gene at different cell type resolutions. And usually, you can see in the subtype level, there's 160 subtypes, so it's pretty dense plot. And by clicking and dragging on the plot, you can zoom in to see exactly what cell type has the lowest methylation level. And let's scroll down a little bit. We see a paired scatter plot showing you the metadata of our cells and this gene's methylation level in the corresponding cells. And here in the coordinates drop down, you can choose all different kinds of embeddings. The level 1 embedding is just like our analysis, is including all the cells, all 100,000 cells in this analysis. So you can have a good overview of all the data we have. And if you this gives you a good overview, but it might not give you too much details if you're interested in specific cell types. One way is you can drag and zoom in to this plot. Excuse me. But another better way, if you know what cell type you're interested in, is might be using a different embedding. For example, by choosing the level 2 UMAP, I'm an excitatory cell, then I'm only showing you here all the excitatory cells, which you can see more clearly that this gene is a marker gene of layer 2-3 and a layer 4 cell, but it also hypomethylated in the claustrum excitatory cells here, and also a small population in the olfactory excitatory cell. And if you're further interested in certain cell type, say layer 2-3, then you can go to the third level TSNE or UMAP. And because this gene is basically a pan layer 2-3 cell type, you can see most of the cell, although there are some populations and not, most of the cell in layer 2-3 are hypomethylated. Okay. And let's go back to the level 1 UMAP. And you can see that you can also plot this by default. I'm plotting you here the major type we get from the analysis. You can choose other cell metadata like the subtype or the brain region, like where the cell is dissected, or many other mapping metrics about the cell coverage and their global methylation level. And also for genes, as I said, we have both CH and CG methylation. Usually for neuron cell type markers, the CH methylation has a broader diversity range scale, and it's usually more cell type specific, but you can also check the CG, and you can see the CPG methylation of this gene is also specifically hypomethylated in this cell type, but the range is smaller. And you can click the scale bar here to adjust the range of the color bar to a certain level that might more clearly show you what it is. And usually for all the neuron cell types, using CH might give you the best visualization. But for the non-neuronal cell types, for example, this gene MBP for the which is a marker gene for the oligodendrocyte, you might want to use the CG methylation level and check the non-neuronal cell embedding, so you can more clearly see that this gene is a marker of the oligodendrocytes. Okay, so this is the cell view of the methylation level of these genes. And in addition to that, we could also plot the cluster level, level methylation profile of this gene using the genome browser. So let's take an example here. The final part of this gene browser will provide you a genome browser that you can plot either the CH, CG, or just the coverage of this gene in different cell types. And here, let's use this for excitatory, cortical excitatory IT cells, which is basically this population, as an example. I selected these cell types, and then I created this browser here. Let me order it a little bit so we can. Okay, so it's basically showing you the same thing, but now we're only showing things at the cluster level, which is layer IT, layer 2, 3 is a cluster, layer 4, layer 5, layer 6, four clusters, 4 major cell types. And then the browser is showing you the same gene, and this is basically the gene body and their different transcripts. And you can see that it's the same result, that this gene is hypomethylated, the gene body is hypomethylated in layer 2, 3, and layer 4, the same as the scatter plot here. And the gene body of the other cell type is much more hypermethylated. And this is the CH methylation. If you go to the CG methylation, you can see that the specificity is pretty much still exists, but CG methylation is the range of differential methylation is different than CH. And usually CH, you can see the methylation, hypomethylation is across the whole gene body, but for CG, the cell type specific hypomethylation usually happens in the promoter surrounding regions, or the intron and downstream and upstream non-coding regions of the gene, where that's why we said you can identify differential methylated regions like this one, like this one, that basically showing a smaller region that is differential methylated in their CG methylation. And hopefully in the near future, we can create a separate browser that can allow you to link, basically annotate all the DMR regions here, and then allow you to click and go to the certain page for specific DMRs and plotting more information about that. Okay, so this is the gene page that gives you an overview about gene information, the cell type level, the cell level gene body methylation level, and the genome browser of this gene at the cluster level. And the next is the brain region browser. We created this table which listed all the brain regions in a hierarchical, sorry, not in hierarchical, basically 45 brain regions we dissected in this study. And each clickable link here will take you to the page of the corresponding brain region. So I already opened a page for the MOP. So this page actually corresponding to 4 dissection region. As you can see, showing in the 3D model here, these 4 trunks are basically we made this model as close as we can to the actual way of our dissection. And in the background, you see the MOP region and the whole mouse brain. And all these are kindly provided open source from the LNCCF. And you can load other anatomical structures for comparison. For example, I'm loading SSP, SSS, MOS. So these are the surrounding brain regions around this dissection. So you might see that we might have some due to the basically we cannot follow exactly the outline of the region, especially in the dissection under the microscope. But here you might see that what kind of overlap we might have of certain dissection region with other anatomical structures. And in the plot below, you can see it's pretty much the same, that you can select a different layout and annotate them by different brain region level, and plot them, and on the other side plot the cell types. And also one thing I forgot to mention here is that this brief intro card also contains links that allows you to navigate through different levels. For example, MOP contains all the 4 individual MOP dissections. If you click this one, it will take you to the page that only has this MOP1 dissection, and the plot below is updated correspondingly. And if you click the isocortex, it gives you a more higher level browser page that basically contains more cells from all the isocortex and similar here. So you can see that the scatter plot is updated corresponding to the what region you selected, and it's only showing you cells. It's using the same UMAP and t-SNE embedding, but it only colors the cells from this region. So you can see that basically the distribution of these cells on our whole brain UMAP. And this sunblast burst plot is summarizing the cell type composition of the cells you see here. You can see we have 80% of the excitatory neuron in isocortex, about 13% of the inhibitory neuron, and about 6% of the non-neuron. But also an important note that this ratio is not the exact ratio of the original brain, because we have fact sorting to select most of the neurons, but only part of the non-neurons, and as explained in detail in the notes here. And if you click certain cell types you're interested in, you can zoom in to see the composition of this cell type as a proportion of the upper cell type level. And also one request you might have is plotting this spatial information together with the gene you're interested. And here in this browser, we didn't provide that, but you can click this link. It will take you to another tool we call the paired scatter browser that basically can showing you can maintain the cell selection from certain brain region, and also adding another plot showing you the gene methylation level, and you can choose any gene that you are interested to plot. And here you can further subset cells. Like by default, I'm coming from the isocortex page. Only the isocortex cell is selected, but maybe you can you're only interested in inhibitory cells. Then you can replot the data by subsetting cells to only inhibitory cell in the isocortex. And GET1 is the marker for inhibitory cell, so you can see they are all hypomethylated. And also to speed up the visualization, all the previous plot you see doesn't plot the whole data set, but we do provide an option here that you can plot the whole 100,000 data set. It might take a few more seconds than the normal plots. Well, not that slow. Maybe I should change the annotation here. But basically, now you see this whole data set. And it looks pretty much the same because the embedding itself is studied from the whole data set. So I think in order to speed up the visualization for especially for some just randomly walk through, it will be good to show downsample the data to show that scatters. But you have the option to plot this high resolution figure image here. Okay, so this gives you the brain region view of the data set. And in the last section, I'm going to show this cell type page. And again, this information card gives you a description and also a navigation to different level of cell types. The one I'm showing here is the layer 2-3, one of the major types. You can go to its parent, which means the upper level of cluster definition is the excitatory cell, or you can go to its siblings, which basically are other major types identified from this excitatory subtype major type cell class. And you can also go to the its children page, which basically are subtypes identified from here. I will stay here and explore the cell type browser. So basically, this sunburst plot is now is showing you the brain region composition of this cell type. You can see it's mainly dissected from isocortex and different cortical regions. And here the bar plot is basically showing you the same thing, but plotted in the dissection region. So this is a number of cells from each dissection region. And then there are some basic information about the mapping quality mapping metrics of this cell type. And then most importantly, we usually need to annotate or show people how we annotate these cell types by plotting their marker genes. And here, similarly, I'm only subsetting to the data to show this particular major type. And the gene plot is showing you all the cells, but with certain genes that might label these cell types. And you can plot any gene you want here. Just import a different gene name. It will give you a different plot. And because this is only showing you excitatory cells, so there's no GET1. Maybe we can go to this is an overall UMAP. You can see GET1 hypomethylated in all inhibitory cells, but not this cell types, which is the excitatory cell, one of the excitatory cell. And in the final part of this cell type page, we actually provided a quick tool to rank the differential methylated gene for the particular cell type. As you can see, COX2 is one of the genes that can distinguish layer 2-3 among all other excitatory cells, but we also provide you many other genes like this gene. If you click the name in this table, it will load this gene on the above scatter plot. You can see that this gene is also a very specific marker in the layer 2-3, although it might have some other cell type hypomethylation also. This is actually a common phenomenon that we see given more and more data that covering the whole brain. We find that it is pretty hard to, if you really want to find a gene that is really unique to one cell type, it is more often that a certain gene is hypomethylated in this cell type, but it most also hypomethylated in several other cell types. So we provide you a more flexible way to identify cell types. If, for example, COX2 is hypomethylated in both layer 2-3 and layer 4, if you really want to find a marker gene that's hypomethylated in layer 2-3 but not layer 4, you can choose any combination you want here in this two drop down. And then it will update the differential methylated gene table. So let's click the first gene. So you can see as we want, this DAB1 gene is hypomethylated in layer 2-3, but very hypermethylated in layer 4. So this gives you a gene that basically is what we selected here. But however, you can see that this gene is also hypomethylated in many other places. You may also, you may be adding more cell types here so that you can rule out these cell types, but the analysis may not always give you what you want because those kinds of, depending on the combinations, those kind of gene may not exist, especially if you really want to find a unique gene that's not hypomethylated in any other cell types. But here we provide a way to prioritize it so that you can explore whether certain kind of gene exists or not in our data set. And the detailed calculation in method used in this table is that it's actually based on pairwise comparison. So we pre-calculated any pairwise comparison of any subtypes in our data set. And then whatever combination you choose here, we use the metric we defined in the manuscript to aggregate the pairwise comparison and to show you the table here. In that case, we don't need to run a differential methylation analysis online. So it is much quicker. As you can see, updating the table only take less than 1 second. Okay. Yeah. So that's it. And I would like to thank all our team members, including our PI, Joe Ecker, and all our collaborators in this project. And thank you very much for your attention.
Ching, this is Joe. Thanks very much for a nice presentation. I just want to point out one thing. While there might not be a unique gene or set of genes for a tissue, there could well be unique regulatory elements that are associated with the expression of that gene only in one tissue, correct?
Yeah, that's actually something we discussed in the manuscript, but because that's definitely possible, and we could you could read more potentially in the manuscript.
Can you go back to the initial slide that you showed that had the to do? Yeah, so the cis regulatory element, maybe you could just add a little bit of flavor as to what you're doing there without going into details.
Yeah. Oh, you mean talking now?
Well, yeah, I mean, just to kind of give a high level overview of the kinds of things that you'd like to see.
Yeah. So one thing we noticed is that some gene is hypomethylated in certain cell type. For example, sorry, I don't have the slides, but we found one gene hypomethylated in 2 cell types. Okay, so it's shared marker by these 2 cell types, but we do find that the DMRs around this gene is part of the DMR is specific to one cell type, part of the DMR is specific to the other. So this gives us an example that the same gene might be regulated by different regulatory elements in different cell types. And yeah, and that's one thing we discussed and could be potentially shown in this browser in the future.
That's great. Thank you very much, Hanqing.
Thank you.
It was a great presentation and a great tool that you built. Michelle, if you can stop recording, we can